So welcome to all the BSS second year students for semester 4. In the last class we have seen that is different aspects of photosynthesis chapter, that is light reactions, dark reactions, C3 pathway, C4 pathway and CAM pathway. Details regarding the light reactions, why it is called as light reactions, why it is called as dark reactions. Then ultrastructure of chloroplast, photosynthetic pigments. Stepwise we have seen all these things. Today I am going to be repeat the same things, whatever the important aspects are there in this chapter, that is in photosynthesis. So while explaining the light reactions, why it is called as photophosphorylation, what is cyclic light reaction, what is non-cyclic light reactions, all these things we have seen very details stepwise. Similarly, the second phase of photosynthesis, that is the light phase, is the first phase of photosynthesis in the mechanism of photosynthesis, and the second phase of photosynthesis, dark reactions, or dark phase, it is also known as dark reactions. Various different scientists, different physiologists have been worked on carbon dioxide fixation, how it is take place, what are the different steps of carbon dioxide fixation. So on the basis of all this investigation, there are three pathways where the carbon dioxide fixation take place, that is C3 pathway, C4 pathway and CAM pathway. These three pathways are mainly responsible for carbon dioxide assimilation or carbon dioxide fixation. So here you have to be again, I have to be discuss step one by one, that is C3 pathway, C4 pathway and CAM pathway. The first thing is why it is called a C3 pathway. In most of the dicotyledons, the carbon dioxide fixation takes place by C3 pathway. C3 pathway. So here, why it is called as so this pathway, first of all, discovered, or the whole process of C3 pathway was given by Melvin Calvin. For the discovery of this pathway by using isotopes of carbon dioxide, C14, for the investigation or the cycle which is proposed, which is given by Melvin Calvin. For this investigation, he was honored with a Nobel Prize in 1962, Melvin Calvin and his co-workers. So here, the first in C3 pathway, the first carbon dioxide molecule is RUDP, ribulose diphosphate. RUDP, which is present in the stroma of chloroplast. As soon as the carbon dioxide enters into the stroma of chloroplast, reacts with RUDP. Therefore, in C3 pathway, the first carbon dioxide molecule is ribulose diphosphate, RUDP. Now, as soon as the carbon dioxide reacts with RUDP, it is get converted into an unstable compound. And now this unstable compound immediately get converted into phosphoglyceric acid. Now this phosphoglyceric acid gives rise to one more reaction, that is 3-phosphoglyceraldehyde and dihydroxyacetone phosphate. 3-phosphoglyceraldehyde and dihydroxyacetone phosphate. So here the first stable compound which is formed is 3-carbon compound, therefore it is known as C3 pathway. Now this 3-phosphoglyceraldehyde, one molecule of 3-phosphoglyceraldehyde and dihydroxyacetone phosphate reacts to each other in the presence of a particular enzyme, gives rise to fructose 1,6-diphosphate, fructose 1,6-diphosphate, fructose 6-phosphate. Likewise, again these 3-phosphoglyceraldehyde and dihydroxyacetone phosphate and fructose 6-phosphate reacts to each other in a particular enzymes used as to erythrose 4-phosphate and cellulose 5-phosphates. Then formation of pseudo plus 1,7-diphosphate, pseudo plus 7-phosphate, ribulose 5-phosphate, and again there is a reformation of RUBP or RUDP take place. So these two important things take place. On one side, there is a continuous formation of sucrose, and by the number of again producing a number of intermediate compound, there is a reformation of RUDP take place, and due to the reformation of RUDP, there is again now it is ready to accept another carbon dioxide molecule and to continue the cycle. In this way, here in case of C3 pathway, there is a synthesis of starch or glucose take place. So this is about C3 pathway. Now, while explaining the C4 pathway, in most of the monocotyledones, there is along with the C3 pathway, there is one more pathway where there is a carbon dioxide fixation take place. That is C4 pathway. In most of the monocotyledones, the carbon dioxide fixation or carbon dioxide assimilation take place by C4 pathway. It is also known as Hatch-Slack pathway. Why it is named as Hatch-Slack pathway? Because these two scientists were given details regarding the cyclic way or details regarding the number of steps which are involved during C4 pathway or C4 carbon dioxide fixation. Therefore, it is named as Hatch-Slack pathway. So here, two important places are there. That is in mesophyll cells, the C4 pathway take place. Some part of the C4 pathway take place in mesophyll cells, and some part of the C4 pathway take place in bundle sheath cells. Bundle sheath cells means the vascular bundles covered with a bundle sheath cells. So in that cells there will be there is a presence of half part of the C4 pathway. So while explaining the C4 pathway, you have to draw one figure of mesophyll cells and another figure of bundle sheath cells. Here in C4 pathway, the first carbon dioxide molecule is phosphoenol pyruvic acid, PEP, phosphoenol pyruvic acids. And as soon as in mesophyll cells, carbon dioxide enters the phosphoenol pyruvic acid, reacts with phosphoenol pyruvic acid, there is a formation of pyruvate and malic acid take place in a particular enzymes. So that malic acid, which is formed in mesophyll cells, transfers towards goes to the bundle sheath cells where there is a decarboxylation take place. Then decarboxylation means the release of carbon dioxide and pyruvate take place. So whatever the carbon dioxide which is released in bundle sheath cells, now this carbon dioxide enters into the Calvin cycle where there is a formation of starch or glucose take place. And pyruvate which is formed during decarboxylation of malic acid came back towards the mesophyll cell, and there is a reformation of phosphoenol pyruvic acid take place. So this is again now this newly formed PEP or phosphoenol pyruvic acid ready to accept another carbon dioxide molecule, and in cyclic way it will be continuously take place. There is a synthesis of starch or glucose. In this way, in C4 pathway, there is a synthesis of glucose or starch take place. There is one more terminology, that is Cranch anatomy as one of the important aspect of C4 pathway. So that is they have to be remembered. So with these two pathways, there is a carbon dioxide fixation take place. That is C4 pathway, C3 pathway and C4 pathway. So you have to remember stepwise all the reactions of C3 and C4 pathways and their particular enzymes. Now there is one more pathway that is known as CAM pathway, cross-relation acid metabolism. Cross-relation acid metabolism. In most of the desert, the plants which are growing desert area where there is a scarcity of water, during day period the stomata are closed, during night period the stomata are open. Therefore, during night period, whatever the carbon dioxide enters into the mesophyll cells, so therefore in case of calm pathway, there is a diurnal changes. Diurnal means during what process take place during night period and what process take place during day period. So during night period, as soon as the carbon dioxide enters into the mesophyll cell, reacts with phosphoenolpyruvic acid, and there is a formation of malic acid. So whatever the malic acid which is formed during night period, stored in the vacuoles, and on the onset of day period, so decarboxylation take place, and now whatever the carbon dioxide which is released under Kelvin cycle, where there is a synthesis of glucose or starch take place, and now there is again reformation of pyruvate or phosphoenolpyruvic acid. So in calm pathway, just you have to remember the important points, important things, that is night period and day period. So what things happens during night period, what things happens during day period, you have to remember. So in this way, these three pathways are mainly responsible for carbon dioxide fixation. So in this chapter, you have to remember certain important points, that is definition of photosynthesis, then ultrastructure of chloroplast, then photosynthetic pigments, what are the different photosynthetic pigments, then what is meant by immersion enhancement effect, what is meant by red drops, then what is meant by Hill reactions, or photolysis of water. Then you have to remember cyclic light reactions and non-cyclic light reactions. So there is a definition. What is the definition of photophosphorylation? What is meant by photophosphorylation? What is the important aim of light reaction? The main important aim of light reaction is synthesis of ATP molecules by adding one inorganic phosphate to the ADP in the presence of light. Therefore, it is known as photophosphorylation. So that is the important aspect. So the main important aspect of light reaction is synthesis of energy-rich compound. So the light energies get converted into chemical energy. And that chemical energy is utilized, which is formed in the ATP and NADPH2 molecule during carbon dioxide fixation or during second phase of photosynthesis. Then after that, you have to give more stress or more importance on what is meant by C3 pathway. So what are the how it is take place, and in reference books, whatever the reference book which I have given, that is a textbook of plant physiology by V.K. Jain, a clear-cut diagrammatic representation of C3 pathway, C4 pathway and calm pathway he has given very clearly, and what are the different steps, what are the different reactions and their enzymes, stepwise they have given. So you have to refer this book for C3 pathway, C4 pathway and calm pathway. So these three things are very important when studying the photosynthesis chapter. Now after that, with this I have completed this photosynthesis chapter. Now the next chapter, which is important for your syllabus, is in this paper, that is respiration or cellular oxidation. In that chapter also, there is a definition of respiration, mechanism of respiration, what is meant by glycolysis, what is meant by Krebs cycle, what are the different reactions of glycolysis, where it is take place, then Krebs cycle, or cellular oxidation, Krebs cycle, then uh, uh, terminal oxidation, RTC, terminal oxidation. What is meant by terminal oxidation? Then how many ATP molecules are produced in glycolysis? How many ATP molecules will be produced in Krebs cycle? So then, what is meant by anaerobic respiration? What is meant by anaerobic respiration? Aerobic respiration, alcoholic fermentation. So all these things comes under respiration chapter. So you have to cover all these things in respiration chapter or cellular oxidation. Now after that there is one more chapter, that is growth regulators or growth hormones. Growth hormones. So what are these growth hormones? What are these different types of growth hormones produced by plants? That is auxins, gibberellins, cytokinins, abscisic acids. So these are one of the most, these are some of the important naturally producing growth hormones by plants for their growth and developments, how they are helpful for growth and development of plants. So you have to be study in details regarding all these things. And the last chapter which is mentioned for your syllabus, that is enzymes one. So with these three chapters or these two chapters will be covered within 6 to 7 periods. With this today I am going to stop here. Thank you and